So I figured that we're going to have people that are going to be talking about clinical data later, how to actually use it, how to access the molecular data. So I'm just going to give you an overview of what you can expect to see out on these public portals. So to start off, just set the ground rules. The Cancer Genome Atlas was started to try to develop a comprehensive atlas of cancer. So they were taking every molecular platform that was available at the time, and so every single patient sample that comes in, it will get gene expression data, will get sequencing data, DNA methylation, gene expression, microRNA, so all that will be available on essentially every single patient sample you see. So and in addition to that, you'll see there's also clinical annotations to go along with that. So you're not just looking at these molecular results in a vacuum. There are certain things that TCGA can do and can't do, so based on some of these criteria. First of all, you should know that all the samples that you're going to see are primary tumors. So that is, we need to have a primary tumor that's de novo, it's not a recurrence or anything like that. These are all invasive cancers. You may see in about 1% of the cases, there may also be a recurrence or metastatic. So you might be able to have what they call a triplet. So you might be able to compare the tumor, the primary tumor, to the metastatic or recurrent, as well as the normal blood sample. Again, TCGA has a list of about 30-some tumor types that they're working with, so it has to fit into those specific subtypes. We try our best to make sure none of these tumors had received any prior treatment. A lot of times we find out after the fact when people are collecting clinical data that yes, in fact, this person did receive a treatment, either for another cancer they'd previously had, or there was some neoadjuvant treatment they received that they didn't know at the time the sample was submitted. This is a genomics project, so we need to make sure there is more tumor present than normal sample, so you can help tease out the tumor signal from the normal. And we also try to have a low necrosis. As I was saying earlier, there will be a normal control, and we prefer blood. Sometimes we will get normal tissue in, sometimes we will get normal tissue and blood. But they really discourage normal tissue, worried about tumor contamination, worried about field effects. So blood is the gold standard. In addition, any of the 150 tissue banks that are participating in the project, they have to provide a de-identified pathology report in English. We do get samples from every continent except Antarctica. So we do make sure those are translated in English. Most of the time we will get a complete report. Sometimes it's just a synoptic 3-4 sentences. We will also ask for them to complete all the required data elements, and then they do have to send in some pathology slides to go along with that. There are some exceptions. So if anyone's interested in melanoma, the primary tumors for melanoma are very hard to find, so the exception has been made. Most of those come in with metastatic only. And there are exceptions to prior treatments. Sometimes standard of care dictates we will not get any samples unless certain treatments are allowed. So, so for these tumor types, we do allow some of these other treatments. So what tumors are going to be out there? So, pick your favorite. Like I said, there are about 35 or so we're working on. These are the ones where we are currently accruing samples. If any of you know people that have a tissue bank and want to participate, you can apply for a contract, submit a proposal. You get reimbursed for your efforts in submitting these samples. These are the ones that are currently open. So Some of these are doing very well. Some of them, due to cancer care, we're not getting a lot of these samples. The ultimate goal is to hit 500 qualified cases. So once things go through our process, we generally have about a 70% pass rate. So we have to get more than 500 patients enrolled on each study to be able to close it out. Most of these, as you will see, a lot of these are carcinomas. We do have one sarcoma study, if anyone's interested in sarcomas. That type, we accept 10 different sarcoma types. So that's a really diverse study that we have for that. And these are the studies that have actually met the accrual goals. And the complete data sets are either out there now or the processing is finishing up for those. Some of these, like the glioblastoma and the ovarian, were part of the pilot study for TCGA. So those were done many years ago, and they've been published on. A lot of these other ones, there are TCGA papers out describing the study sets for the colorectal. And really, that's about it. All the other ones, so glioblastoma, ovarian, and colorectal have published papers that you can find, and they're published in Nature. The other ones, we're working on marker papers right now, so you could probably expect to see those in the next year or so. And then we do have a new project that opened up in the past year. So these are rare tumor studies. These are studies where we will never get 500 patients to qualify. So they took, the goal is to try to get at least 50 to 100 of each of these types. So we have finished the chromophobe kidney. We're currently processing the last of the uterine carcinosarcomas. And which other one are we? The adrenocortical carcinoma. A lot of the others are just starting. So you'll see in relation to the previous talk, mesothelioma is on there. And so we are actively collecting for the majority of these. So again, the ultimate goal when TCGA started back in 2006 was to look at cancer and try to get as much data as is possible based on our current technologies. So a lot of that includes some of the patient information. You can also see there's pathology information. There's a lot of molecular information that we collect. And at the very end, we also try to get proteomics data when feasible. So that's more of an add-on, and you'll find some proteomics data here and there, but that's generally not for every single study. So again, these, the three tumors you see there were the original ones in the pilot. To talk about some of the difficulties, I said this started in 2006. The squamous cell lung carcinoma, we received our last sample for this last month. So we have been accruing that tumor type for 6 years. So now to get all this data requires a large team. So TCGA is made up of centers all over the country, and we have one site in Canada as well that participates in these studies. I was talking a little bit about what molecular data you can expect to see. So the middle column refers to the platforms they were using in the pilot study. During that time, when they started the pilot and then they went into an expansion phase in 2009, the cost of sequencing just dropped precipitously with the introduction of next-gen sequencing. So some of these technologies went away and were replaced with sequencing. We retained the SNP6 panel. We retained the DNA methylation. And a lot of the others, you can see that RNA sequencing technologies are there, and we have DNA sequencing. If you're looking for DNA sequences, what you can expect to find is whole exome sequencing. So every single case that goes through the pipeline, you will get whole exome. Approximately 10% of the cases are selected for whole genome. So since the exome makes up such a small percentage, it's a lot quicker, cheaper to be able to do the whole exome. And since most of the things people are looking for are in coding regions, you get the most bang for your buck. But you will also find there are whole exome sequences out there on some of the more mature studies. And again, proteomics has been introduced, and there are generally two modalities. So the reverse phase protein array, you will have more data on that than on the mass spectrometry. So again, here are all the centers that are participating. The blue is me, so coming from Columbus. So even your cold snap you've been having here is still a lot warmer than back there. So we are what they call the Biospecimen core resource for the project. So we are the ones where all the samples come in, we do all the initial QC, and then isolate the DNA and RNA, and the DNA and RNA then goes to the centers, the sequencing centers, which are in green, and also to the characterization centers that are in red. There is a data coordinating center, so every bit of data that's generated from these samples, it will eventually reside at the DCC. So what we do at the Biospecimen core resource is we make the initial contact with approximately 150 different tissue source sites, they call them, tissue banks, but in the TCGA lingo, they refer to them as tissue source sites. So the first step we do is we have to go through all the regulatory documentation, make sure that, that the centers that want to submit the samples actually have IRB approval to do it, and it's consistent with the consent of the patient. We provide them a lot of training. This is a project like no other, and we make a lot of demands on the sites that are participating. There's a lot that we ask for to try to get the most complete set. So we have a large, what we call a clinical outreach group that is our kind of outward-facing customer service area. They spend a lot of time training these sites, both in using OpenClinica, which is our web-based clinical data system, and also training them as to what we're really looking for on some of the clinical data questions, ensuring we get the right samples in, make sure they know how to ship a cryoport, which is somewhat unusual. It looks like a little mushroom, but it's filled with liquid nitrogen vapors. Most people are used to shipping on dry ice, so we spend a lot of time working with the tissue source sites to make sure we get the best samples, best clinical data in here. The next step, once we get our first shipment in, we will go through and start doing our pathology verification. So the site is supposed to pre-screen all their cases. So they take the frozen tissue they want to send to us, and they do a pre-screen to make sure that it has a sufficient amount of tumor and the right diagnosis. We verify that at the BCR. So we will go ahead and cut our own frozen section. We have our own pathologist that will go review, make sure the diagnosis is consistent. We also look through the pathology reports, make sure there's nothing that looks odd. If we have those kind of questions, we go back to the site and make sure those are answered before we even send them into the lab. So that's our first quality control step. The ones that actually that pass through our pathology QC then move into our molecular laboratory. We have a co-isolation procedure, so we will take one piece of tissue. From that piece of tissue, we will cut a frozen section. If that qualifies, then that exact piece of tissue goes into the molecular lab, and we homogenize, run it over a DNA column. isolate the DNA, and then all the flow through goes over an RNA column. So what you are seeing is we are doing a review on the exact same piece of tissue that is processed and data generated from. That slide eventually ends up at the DCC as well. So if you're looking at some molecular results that look kind of odd, you can actually go and pull up the slide and have a pathologist look at it and say, okay, yeah, that seems right. So we put all that out there. Once we get the DNA and RNA, it goes through a rigorous quality control. We have to make sure the RNA is of very high integrity. We do an identity panel. So we double check, make sure that the tumor and the normal are from the same patient. So, and about 1 or 2% of the time, that's not the case. And then we have to kind of resolve it. But if you are sending these samples out for gene expression, sequencing, DNA methylation, you're talking about thousands and thousands of dollars. And so we'd rather just stop it right here before all this money's wasted. So then we distribute these nucleic acids to all the centers, and then they start working on it, and then we turn our attention to the clinical data. We spend, I would say, half of the BCR's efforts are in actual processing of the physical samples, and the other half is dealing with the clinical data. It is a huge process. So rather than having everybody submitting all the data up front and spending a lot of time and then turning out the case doesn't even qualify, we wait until all their samples are processed. We send them a report and say, okay, we're now ready for your clinical data. And that's when they start going through that. So basically, this is a brief description of how it works. So the center part is all the specimen processing, and then clinical outreach is they're the group that really handles all the clinical data. So this is what I'm going to spend most of the rest of my talk on. So people may be wondering where the clinical data comes from. Who asks the questions? Who determines what's important? So very early in a study, some experts, both within TCGA and outside experts in that tumor type, will get together and they will form a disease working group. We have some generic forms that are available, and we share those with the group and work back and forth deciding, well, are these questions relevant to this cancer type? Are there any other cancer questions that are really important that you feel we should capture and we should be able to get? So we work back and forth with the disease working groups over many iterations to get the clinical data form in place. Then we work with the Cancer Data Standards Registry, so that's part of the NCI. We will actually then register these questions with the CADSR. So generally, there are a lot of these clinical data questions that are being asked for other projects in the National Cancer Institute. And they may already have specific questions laid out and very defined answers. So we will generally review to see if people have already asked these questions and registered them or not. And if not, then we will go ahead and register them with this agency. So if you want to see if somebody's asking a question and you know the question, you know every single choice that's allowed for that. So, and it's consistent across tumor types. So if we're asking a question for the cervical cancer study, we're asking the exact same question with the exact same answers for the endometrial study. So way of standardizing all this. Once we have gone through that process, we have the final forms. We develop it in Open Clinica, generally have some tissue source sites do some testing, and then we also will start developing our XML, and I'll talk about XML in a little bit as well. So that's our main way of actually uploading the clinical data to the Data Coordinating Center. So this is how the forms start off. So we start off with just PDFs. So we call these our wireframe forms. All of these forms for all of our studies are available on our website. So if you want to see what the tissue source sites see when they're filling out the questions, you can go to our website, freely download them. So I mean, these are paid for by government funds, so we try to make them publicly available. So we start off, like I said, with the generic form, do an electronic build, and then we generally start collecting data. One of the issues we find is when a tumor study tends to become more mature. So once we have about 200 or so tumors that have qualified and shipped, then an analysis working group tends to form. And they look at what data has been generated, and they look to see if there's anything interesting out there that hasn't been discovered yet. Any new correlations they find, new genes that are of interest, new microRNAs that may play a role. And a lot of times that's when people first start looking through the clinical data. And that's when people start saying, it's like, oh, well, we also want to get this information. And at that point, we're like, well, these sites have already processed 200 cases, and they're not going to go back into the patient records, pull all the data out. So there are some data elements that people realize, a year or two later, oh, I wish we'd asked that. So, but I think that's just the nature of doing this kind of research. So here are the different kinds of forms that you will find. These are the ones that we ask. So with every sample that is submitted, the sites will complete a case quality control form. So that is essentially just information about the sample and a broad information about the patient, such as the histology, might include the amount of tumor nuclei they found or the weight of the sample. So some just very basic information. Once we go through and we process all their samples, send them this report back saying, okay, these qualified, we need new data, that's when they are required to complete an enrollment form. So they're contractually obligated to complete these forms within 60 days of receiving notification. It's the enrollment form where you will get the most data about the patient. So that goes into, did they receive treatment? Did they have recurrences? If there's some specific information about that tumor type that they're trying to capture, it would be in there. So a lot of times we actually send the sample out and it's starting to be processed, and we might even have some molecular data. We don't even have the clinical data yet. So there's a little bit of a disconnect for that, but eventually it catches up. We are also requiring all the tissue source sites to do 1 year of follow-up. So 1 year after we get the data from the enrollment, they will follow it up with the patient to see if they're still alive, if they've had any recurrences, are they disease-free, et cetera. So that, again, will kind of be delayed. So some of the more recent studies, we don't have much follow-up data on. So those forms are required by the contract that all the tissue source sites work under. The other forms are optional. So sometimes you will receive information on what treatment they received, be it pharmaceutical or radiation. We also have an ablation form. So for the liver study, sometimes they're treated by radiofrequency ablation. And if the patient did have another cancer at some point, we want to capture that information. So we also have another malignancy form. So if they'd had colon cancer 10 years ago and then they're presenting with breast cancer now, we want to capture that, yes, they had another cancer previously, just in case that's important. Also, if it's a synchronous malignancy, sometimes you can have bilateral breast cancer. So if we get cancer from one breast, maybe there was a tumor in the other. So we want to note that information as well. These are on the enrollment form, these are the minimum data elements that are required. I'm not going to go through all these, but these are required for all sites to complete. And I can share this presentation with you guys later, so you don't have to. So if there's information you're interested in, I'll make sure this presentation is available. So this is on the enrollment. We have more limited data on follow-up forms, but again, pertains to how did they do after treatment. So you can better use this to calculate, survival curves and whatnot. Now, I did talk about, some tumor-specific questions. So, we do modify the required elements, and then we do add a couple of additional required elements depending on what the tumor type is. So, obviously, if you're looking at lung cancer, you're going to want to know their smoking status. ER, PR, and HER2 positivity is very important in breast cancer, et cetera. So these data are collected as well. I did talk a little bit about how we try to standardize. So if we are using one question, we make sure it's the same. So the big push for standardizing the clinical data to the greatest extent possible is because of a new group that's formed over the past 6 months in the Cancer Genome Atlas, called the PanCancer Group. Because we are processing every single tumor type the same way, we are sending out samples to all these characterization and sequencing centers, and they are doing the exact same processes to generate their data. What you're doing then is the breast cancer samples you have are processed the same way as the endometrial cancer samples, as the colon cancer samples. So you can actually do cross-study comparisons. So what this group is trying to do is they're looking at the study sets, and they're ignoring the tumor type, and they're trying to see if there are any signatures that are common across cancer types. So that if you do see a mutation in somebody that has a mesothelioma, and you also know that that's implicated, in CML, that, maybe we can make those discoveries ahead of time without having to do it kind of ad hoc. So to support that group, we are trying to standardize the clinical data as much as you can with clinical data. We also do some ICD coding. So we do coding both by the site and also histology coding. This is what takes a huge amount of effort at the BCR, is making sure the data makes sense. So we've developed, to date, I think we have about 150 automated validations that just kind of run nightly to check for things like, okay, the date of diagnosis has to come after the date of birth or before the date of death. These are things that every once in a while it gets flagged because somebody just types in the wrong number. Or there are, and we're even trying to get more specific. For instance, in thyroid cancer, you can stage a cancer, you stage it differently if the patient is under 45 versus over 45. So if you see a staging that applies to someone who's over 45 but they're only 30, then that should send up a flag as well. So we have a lot of these validations we're trying to build automatically. So that's our validation engine that we have. When they're actually entering things in Open Clinica too, there are a number of validations to prevent them from entering text in a numeric field, for instance. So those are the automated ones. Then we do a manual review. So currently we have 3 certified tumor registrars working. They review every single case. So they go through all the data and they see, based on their experience as tumor registrars, does this make sense? Does this make sense? And then if it doesn't make sense, they come up with their list of questions. We send it to our outreach specialists, and then they coordinate, communicate with the tissue source site and just say, hey, we had some questions about these. Are these correct? And sometimes they are. We just have to check, and sometimes we have to update it. So we go through a lot of these processes. So once everything has been cleaned up, so you can see over on the left-hand side, the paper forms get implemented into our Open Clinica, and then the questions, once they complete it, they go into our data warehouse, and then we generate XML. So I don't know, most people in TCGA do not use the direct XML files. If you are a computer geek and really, the bioinformatics folks, I'd recommend going to the XML. That's where most of the data reside in, and we actually build it into the XML in a hierarchical structure. So XML stands for Extensible Markup Language. Trivia fact for the day. So we use this for transmitting all of our clinical data to the Data Coordinating Center. So as I said, we define the structure of it. And then each data element has different tags, and they're kind of defining what the different elements are. So if this looks scary to you, then this is probably not for you. If you're a computer person, you might get really jazzed by this. But so this is like one question and all the data that's available on that. So we know, I'll give you some definitions here. So the data name, so basically what the question is. If it's registered, what the registration code is. So you can actually take that code, go to the CADSR browser, type it in, and it comes up. Tells you exactly what it is. So we have display orders. So one thing that we're working with the DCC to fix is we have all these questions laid out in a very specific order on the forms. So for instance, if you're talking about, if you have a question, date of death, you're going to have, one data element's going to be the date, the day field, one's going to be the month, one's going to be the year. And we might ask for some other dates as well. One thing, if you want to peruse the data, sometimes they were just put together, and they were put together in alphabetical order. So the date of diagnosis, so it would be the day for the diagnosis, and there might be another question, date of death or whatnot. So those 2 days would show up side by side, completely out of context. So we are working with the site to actually display them in a proper order so they make more sense to the end user. We're not there yet. I'm hoping in the next month or so that'll be resolved. We also have a preferred name. So we might have a long string that's in our database for what the data element is, but if you want a human readable thing, so we capture all this information in the XML. We have 2 different XML files. So one is the Biospecimen XML, refers mostly to a lot of the information about the biospecimen. And then the clinical XML is much more detailed. Most people, if you're going to look for the clinical information, you're not going to look in the XML. You're going to look to what the TCGA refers to as BioTabs. So the DCC gets our XML file, and then they just chunk it up into bits, and then they dump it out into a text file. So it's very kind of brute force. So down here is what a typical one looks like. So you'll have the name of the question at the top, and then you'll have the answers down here. So you can open this up in Excel, and then it's essentially a spreadsheet. So this is how most people work with it, but there are certain caveats to it. You do lose some data by looking at it this way. And like I said, all the date fields are right next to each other, and it takes a little bit of reordering to make it more useful. Like I said, we've been working with the DCC and to get them to try to change their processes, so this is more user-friendly. I have a couple of handouts I will also distribute with this presentation to everybody. I have them electronically, so I'll ask May to send an email out. how to access these. So there's where to go to, how to parse through the file structure to actually find these data. So if you want to access them, the vast majority of the data for TCGA is public access. So you can go, like even right now, and just go to the site and go download it. So even to the level of the raw data off from the RNA sequencing. We have like the clinical data out there. We have pathology reports out there, and most of the molecular platforms. Like I said, pathology reports, we have these whole slide images. So we have both the image we used to qualify that case, and we also have a higher quality diagnostic slide that comes from the paraffin embedded tissue. So the site will send that to us as well. So more pathologists would prefer the formalin fixed one. For a few tumor studies, microsatellite instability is an important factor, a prognostic factor. So at the BCR, we have done MSI testing for, I'm pointing out some of these codes right here. We have COAD and READ. Those are TCGA codes for colorectal adenocarcinoma and rectal adenocarcinoma. I've got a cheat sheet at the end of this presentation. So we performed MSI testing on all the colorectal cases. We're just starting to do for the gastric adenocarcinoma cases, or the STAD. So we're working on that as well. So we've started, we're working on an assay right now as well to do HPV testing. So this is for the head and neck study. They were able to pick up some HPV sequences during their sequencing runs, and so we're running another assay to validate those results. Again, I'm using a code at the bottom for the cervical study. So HPV is very, the major risk factor in cervical cancer, so we'll also be doing HPV testing. This should be able to give us even which subtypes are involved. So. Again, here's what a typical pathology report will look like. We will redact any of the patient names, but there's a certain amount of information we're really trying to ensure is captured. We also do the ICD-03 coding, so that's generally handwritten by one of our tumor registrars. Again, if you've never worked with whole slide images, those are freely available. They're generally about 2 gigabytes in size, so make sure you've got a good bandwidth connection. But we scan them at high resolution, and you can peruse these at your leisure. Now, there is a controlled access site. So the data that are being generated are potentially identifiable. So especially the SNP6 raw files will give the single nucleotide polymorphisms for a patient. So we don't just do the tumor, we actually do the normal too. So that is essentially the person's fingerprint. So we don't make that publicly available. You have to apply for access. You have to have some IRB approvals. It goes through, and then you're available to download that. Same thing with the raw sequencing data. So those are referred to as the level 1. So you can generally get the level 3, which is like the mutation calls. So you in the public access. So if you're just looking for what mutations have been identified, you can just go to the public access. If you want to actually do your own analysis on the raw files, you need to get into the controlled access. I have instructions on that too. Now, anyone who has worked with human samples, human patients, there are those times when things just don't fit. And trying to make everything go well, it's just an impossible task sometimes. So there are certain times when we may discover when the site's submitting an enrollment form, it's like, oh, we missed this earlier, but they did have a treatment. So, but generally the analyses have already been done, and we're not going to we're not going to pull back those data files. Those are still public. But you'd want to go to this annotations manager, which is also on the main TCGA data portal, and you can look up the cases in question. And you can see if there's anything funky noted about that. Some cases have been redacted. There have been, I believe there have been about 4 or 5 patients in TCGA that have withdrawn consent. So if the data have already been made public, there's nothing we can do about that. But to prevent further data from going up, we will put an annotation saying the patient withdrew consent. So there's a lot of information in there. So if you find something really exciting in the data, you want to go through and just clarify here that there's not something else you're missing. Now this is something that everybody who's in TCGA understands really well. So I put this cheat sheet here for you guys. So the infamous TCGA number. There's a lot of information here, and all the analysis groups just kind of throw these numbers out, and they're fairly comfortable with the sequence. The first 4 digits are just a throwaway. Every single sample will begin with TCGA. The next 2 digits refer to the tissue source site. So what site actually provided the samples to us? So that can get kind of complicated, because if a site at, say, MD Anderson Cancer Center, say they're sending us bladder cancer. And then there's another investigator at MD Anderson Cancer Center that's providing us with the cervical cancer. They will have different codes. So this code is dependent on, so it's unique to both the principal investigator of that tissue bank, the location of the tissue bank, the institution it's at, and the tumor type. So you could have one PI who runs a large bank sending 10 different tumor types. He will have 10 different tissue source site codes. But these are actually, you can decipher which site these came to. So I know Rayhan really uses this information because he's worked on batch effects. So trying to see maybe all the breast cancer cases from one site are being skewed a different way because maybe they process them in some way that is really not right. So sometimes if you're having problems, you can look and see it's like, well, is it isolated to this one tissue source site? And we were actually, there's some suggestions even in one of the studies that there are differences depending on which country they come from. So we're starting to see that. And you can kind of figure out really quickly just by looking at that. We also have the next code is the patient ID. So every patient will have a unique identifier. After that, the sample identifier. So it kind of tells you what kind of sample you're looking at. So the 01 refers to a primary tumor. If that's a 10, that refers to blood. 11 refers to normal tissue. So these definitions are out there. They're not terribly easy to find. But I can help you figure that out. Some of the bigger ones I pointed out at the very bottom. So solid tumor is 01, blood, et cetera. So the vast majority will be those tumor types. The others, generally people are not interested in. So it refers to which actual portion of tissue we cut. So if we get a sample in and we cut 3 pieces, which one of those pieces went up? For the most part, you're not going to care about that. Which plate was it on when we shipped to British Columbia? What was the plate identifier for that? Generally, you're not going to be interested in that. And then the center identifier, that refers to actually which center. Did the data come from University of North Carolina? Did it come from the Broad Institute, et cetera? So that is the number. So generally, earlier, the left half of the number you're going to be interested in. There are also unique identifiers that I'm not going to go into. So every single data element that is available has a unique identifier, what we call a UUID. It's a 36-digit alphanumeric thing. It's intended only for computers. It's not meant for human consumption. So I'm not even talking about that. But if you see UUID somewhere, you can just block it out, because it's, unless you're into computers and you're bringing in large data sets and you're going to do some analysis on it that way. So now here are the tumor codes. So again, early in the project, it was decided that every tumor needed some kind of code. And so these are kind of defined by TCGA as we go along. So in general, the first part of the code refers to the organ where it comes from, and then the second part would refer to what type of cancer. So if it's the bladder carcinoma, BLCA, we have, let me see if I can find, if you look at, let's look at the lung, so that's kind of a little lower down. So we have lung adenocarcinoma and lung squamous cells. So we have LUAD and we have LUSC. So there are these kinds of codes out there too. So somebody new coming in, generally you can figure these out. But again, I just wanted to make these, present these in case you had questions. The one that always gets me is pancreatic and prostate. I always have to do a double take on that one. Again, I'll share these with you in case you come across these so you know what they are. I would be remiss if I didn't point out that TCGA has a publications policy. This is updated on the main TCGA website. So essentially, we're doing a lot of work. We have 7 data analysis centers that are actually, so once the data are generated, then we have groups that are going through and doing analysis on this, and actually trying to find associations. So part of this is allowing TCGA to review all the data first and give a very broad overview. What we find, what do we see in these data sets? So for every tumor study, TCGA will publish a marker paper. I mentioned earlier there have been about 3 or 4 that have been out. They do put a moratorium on anyone publishing from these data sets. So it starts once we've hit 100 cases. Once the BCR has shipped 100 cases out to all the centers, then the clock starts ticking. So 18 months after that date, then everything's free game. So for these slower accruing tumors, sometimes we hit 100 cases, and then 18 months go by and we still don't have enough to do a good marker paper. But then the moratorium's lifted. Then it's fair game. Anyone can go ahead and publish whatever you want on it. If there are some data that you want to publish on, generally the rule has been if you're only working with one platform, so if you're only working with the DNA methylation data, if you're only working with the gene expression data, generally you can go ahead and publish on that. So the moratorium doesn't apply. If you're using multiple platforms, comparing DNA methylation to the gene expression, then usually the moratorium is in place for that. So again, this is out there. If you have questions, there's an email there, and try to get additional permission or whatnot. I do know Dr. Kenna Shaw is the director of TCGA, and even though she gets over 500 emails a day, she is generally very responsive, kind of scary, kind of in a scary way. So I don't know how she does it. I don't get as many as her, and she beats me to the punch a lot of times in email. So, and she is really oversees the publication policy. So if you have questions on that, feel free. Again, if they may already be out of moratorium, or as soon as they publish a paper, then the moratorium's lifted. So even if it's like you're only 6 months in, then it's lifted. You can go ahead and publish. On the website, they also have a list of the tumor studies that currently have a moratorium in place. And again, so here is the place where everything resides. The main TCGA data portal, so all the information's there. And I don't know if I had anything else. And again, my little plug, if you know anyone with tissue samples, we'd love to have more in the project. So, okay.